Okay, maybe we should just get going and people join as we go along. Yeah, good afternoon, good evening, good morning, everyone, wherever you are. My name is Stoyan Stoichev from the Resin Bio team, and welcome to another episode of Emerge. I'm excited today to host Dr. Christof Lenz. Before we get going, just the usual housekeeping announcements. We are recording the session, we're going to make it available via YouTube channel, so you can view it at your own leisure later. Please post your questions, Q&As window or the chat window. We're going to have plenty of time to get to these after the presentation. Also, if you want to have your bit of a discussion, you can just use the raise hand icon and activate your mic. This will be great if you can just have a bit of a chat afterwards in terms of the work that's going to be described. So Dr. Christof Lenz, he's currently the director of the core facility at the University Medical Center of Göttingen, and also a staff scientist at the Max Planck Institute for Biophysics, again at University of Göttingen. And Christof today is going to be telling us about some of the work that his team is supporting in cardiovascular research, specifically on some of the workflows being developed to profile different cardiac tissues to do a proteome and phosphoproteome profiling. So Christof, first of all, thank you very much for coming on board to present your work, and I look forward to your presentation.
Yeah, thank you, Stoyan and team, for the invitation and the opportunity to present our work here. I would, as the title suggests, like to talk to you about our efforts for running mass spectrometry-based proteome analysis in cardiac tissue samples specifically, which is an approach that is very dear to us, as we work very closely together with our colleagues in cardiac research or cardiovascular research. And we see quite an opportunity there to profit from establishing modern methods in proteomics mass spectrometry. So to kick off, some general background. A lot of the focus in cardiac research, but also in other levels of biomedical or clinical omics research, is currently on genomics and transcriptomics. However, the reason why we also would like to look specifically at the proteome and at proteomics is that disease risk is not something usually, or not always, that is actually given to you by your genetic makeup. Sometimes it is due to mutations, sometimes not. And especially the non-genetically encoded disease risks or factors accumulate over your lifetime, which basically is reflected by the fact that a lot of the widespread diseases, heart disease, cardiovascular disease, cancer, have their main onset in the 40s or 50s of people's lives. And therefore, we think there's quite some value in looking at the proteome for disease causes or disease diagnostics. And this is a little scheme I borrowed from Jenny van Eyck, which I think illustrates this very, very nicely, that especially for these late onset widespread diseases, the proteome may offer quite some value for diagnostics and disease understanding and potentially also treatment. Some of this is also reflected, or the acknowledgement that this is largely tissue specific is reflected, for example, by efforts like the Protein Atlas, which is largely run out of Sweden, which offers an antibody-based compendium, you could say, of the human proteome, and specifically of protein expression across many human tissues. And if you analyze or look at this resource, which is very educational, you quickly recognize that the proteome is largely tissue specific. And a lot of the research has been devoted to cancer research and to cancer cells, cancer tissues from biopsies, for example. A little less so actually for cardiac research. Part of that has to do, I think, with the fact that cardiac samples, biopsy samples, for example, are not as easy to come by. For example, if you work in oncology, it's pretty standard procedure for diagnostics to take biopsy samples. The only reason when you should get a cardiac biopsy taken is actually when you already have a heart operation. So, and even then, this is not currently not routinely done. And this is one of the reasons, plus others, I think, why cardiovascular research and specifically cardiac proteome research are maybe a little less developed than the corresponding fields in oncology. So, which kind of tissues do we cover here at the University Medical Center? This is a rough overview of the types of tissues that we have started, or body fluids that we have started looking into over the years, over the last 5, 6, 7 years. Where we have tried to establish routine mass spectrometry-based proteomics workflows for a number of tissues based on the demand coming here from the colleagues, the medical colleagues, surgeons, medical departments that we are collaborating with to do proteome analysis. And as I said, today we are going to focus specifically on disease research in the heart, in the human heart specifically, with regard to atrial fibrillation, with regard to aortic valve stenosis. And there's one thing which I will also refer to towards the end of the presentation, to so-called phospholambanopathies, which is a genetically encoded, reasonably rare, but increasingly well characterized and well recognized subtype of cardiac disease. So, cardiac disease is a relatively recent discipline or widespread discipline, which you can see by the fact that the first cardiac proteome coming out of the group of Matthias Mann in Martinsried in Germany was only published around 4.5 years ago in 2017. And this is really the seminal paper on this by Sophia Doll and co-workers from the group of Matthias Mann, who analyzed or who tried to perform, and I think achieved, a comprehensive proteome characterization of the human heart. They took 3 healthy human hearts, and actually for proof of principle, also 3 human hearts from patients, or actually from deceased patients with atrial fibrillation. And they did a comprehensive analysis by looking at 16 different heart regions, doing a high level of fractionation. They also isolated different cell types, like fibroblasts and so on, endothelial cells from different regions of the heart, and performed a really in-depth analysis of the heart to achieve a complete catalog of the protein expression complement of the human heart. And you can see the numbers here. In any of the human heart, chambers or in the valves, you can achieve identification and also quantification, if you spend a lot of effort, of around 8,000 proteins, or expressed protein species for each of the locations of the 16 locations. This is very impressive work and extremely valuable resource. However, the downside is this took around 3 months of instrument time. If you read the publication and do the numbers, basically they spent 3 months of instrument time to catalog 3 human hearts. And of course, our goal is to see how we can make use of this and get this very comprehensive approach into an approach which is more routine, where we can quantitatively compare higher numbers of samples to perform clinical studies, actually. There's a lot of benefit also still from this in-depth study of the Mann group. Besides only being a stem collection or catalog, they also provide extremely valuable information about localized expression of proteins in the human heart, in the cavities, in the vessels, in the valve, about the different abundance ranges of proteins. For example, as you would expect, the active species or active cell type in the human heart is the cardiomyocyte. And a lot of protein mass, and actually therefore the proteome, goes into the myofibrils, who perform the actual contractile action, but also in energy generation in the mitochondria. So this is very, very useful background information, which actually we have started whenever we look at in-depth at our own data sets. We use this information from this reference publication as a background. So how do we now go about it? In principle, we have switched to a different mass spectrometry approach, that is to data-independent acquisition mass spectrometry. Why data-independent acquisition mass spectrometry? It's a label-free approach because it's actually quite difficult to label human hearts or even mouse hearts for that with isotopes. Of course, you could perform chemical labeling by TMT, but we decided to forego that because we really want to be able to do serial analysis. So not just of the 10 or 16 samples that you can include in a, say, TMT-based experiment, but we would really like to perform studies of NS50, NS100, NS200. And the road forward to this is data-independent acquisition as a label-free quantitative mass spectrometry technique. And contrary, I won't go into this in a lot of detail, to the sequencing-based DDA or data-dependent analysis mass spectrometry on the left side, DIA does no longer select for individual peptides to sequence, but actually takes or subdivides the whole peptide population of a digested human proteome sample into windows, formerly called swaths, and co-fragments inside these windows all peptides. And the information of the identity of the individual peptide and also its quantity is then extracted from these highly convoluted data by typically using annotated MS-MS spectral libraries. So if you have a spectral library with known or previously detected and fully sequenced peptides or PSMs, peptide to sequence matches, rather, you can use this to extract quantitative profiles for all of the included peptides from your DIA-MS data. Increasingly, actually, you can also do this without. This is an approach called DDIA or direct DIA. This is due to software and bioinformatics improvements where you can actually forego the use of a pre-established spectral library. And this external information, this library information is used mainly to control actually for its discovery rate. Because due to the highly convoluted nature of data-independent acquisition, for its discovery rate initially was a very big issue. And the library is actually used to control for its discovery detections of the peptides that you use to quantitate. And in turn, when you can quantitate the peptides of a protein, you combine this to a protein quantity value. So DIA-MS, it is for us. And this is the approach that we use for routine label-free proteome quantitation. Let's see, what does this data look like? On slide 8, top left panel, this is from the analysis of complex cellular lysates actually, but just for reference, I use these. On the top left, you see total ion chromatograms. This is full proteome, cellular proteomes, separated over the chromatography retention time of around 1 hour. So including re-equilibration, this is like 80-85 minutes per injection. And then you can appreciate from the total ion chromatograms that we are looking at full complexity cellular full cell proteomes. And from this very complex profiles, if you have the identity of the peptide and a set of sequence-specific fragments, like in the top right panel, you can then extract traces. So for example, for this peptide with the LIVK sequence, which is depicted there, in the very complex DIA-MS spectrum at any given time, we only detect relatively small, as you can see in the top right panel, a few sequence-specific fragments of interest. And these don't even need to be the highest or most abundant fragments. However, if they are sequence-specific and we extract their intensity over the chromatography retention time, which you see in the bottom right, then we can extract very clean chromatographic profiles for these sequence-specific ions for this peptide. And then we can integrate the area underneath these extracted ion chromatograms, or XICs, and use this as a proxy for the quantity of the peptide and the underlying protein across many samples. In this example, which is the bottom left panel, the intensity over 80, not replicate actually, but 80 individual sample injections. And this is basically how DIA works and is a nicely established routine workflow nowadays. So how does this look like in practice, or what does it look like in practice? Coming back to cardiac research, we take cardiac samples. Tissue samples is what I'm going to be talking about today. And we take small tissue aliquots of somehow where the size of 1 or 2 pinheads actually. So a few milligrams of wet weight of tissue. And we lyse it by a technique called PCT, or pressure cycling technology, which I'll come to in a minute. We digest the proteins after the lysis of the tissue with trypsin typically. And then we run our swath, which is the older Sykes marketing name. The technical term now is data independent acquisition mass spectrometry, or DIA-MS, to generate quantitative profiles. And to obtain a library, the spectral library that I talked about a little earlier, what do? We take from this middle main workflow, we take aliquots of each sample and pool them together to do a sort of representative or average sample. This we pre-fractionate using reversed phase HPLC fractionation at basic pH. We acquire it by data-dependent acquisition mass spectrometry, or DDA-MS. This is the part on the left side, and sequence the heck out of it, literally speaking, identify proteins and generate this MS spectral library. So a set, or as comprehensive as possible, set of annotated MS spectra for sequence, for peptide sequences. And then we use this MS spectral library and use it as a comb to extract these extracted ion profiles for as many peptides as we can from the DIA or swath MS datasets. And then when we have this can be a few 10,000 of peptide extracted ion chromatograms, and have the areas, the quantitative values derived from this. We can do some quality control and then perform statistical analysis. And this is our old equipment and workflow, which we started out with in cardiac tissue around the year 2016. So I'm going to show you the first data set, which actually highly motivated me to follow up on this. There we used an exogen nano-LC, Sykes TripleTop 5600, which was then our standard DIA mass spectrometer in the facility, and the then current software packages. How about this PCT? What does this look like? This is an instrument that we use for this called a barocycler, which is manufactured by Pressure Biosciences and which works very nicely for this. And on the top left, you can see this. We have small plastic tubes, which are roughly the height of a 600 microliter Eppendorf. And there we put in these very small, I said, like a single or 2 single pinheads of tissue. And we seal it with a pestle. This is, if you go to the top right view, this is a close-up view of some of this mushy red cardiac tissue. And the white thing which you see going down is this pestle. And then these closed vials with pestle on are actually exposed to cycles of very high pressure, like 45 kpsi, 0, 45 kpsi, 0, 45 kpsi, 0. This is why it's called pressure cycling. And if you do this long enough, you get a lot of mechanical stress and internal heating also into the sample. And this makes for a very efficient tissue lysis. So it breaks up the tough cardiac tissue very nicely. Actually, you can do even up to cartilage tissue. So you can even do tougher tissues efficiently with this. And also the internal energy that you put in accelerates tryptic digest. So once you put in the trypsin into these vials, you can actually perform a complete trypsination within 60 minutes. So all this speeds up things, and you can do it for 16 samples in parallel. This is again moving to the top left picture where you see this barrel type structure, which looks like something taken out of a revolver. This is the housing for 2 × 8 samples, which can be processed in parallel. And going from a small tissue, which is actually 1/2 or 1/3 of a puncture biopsy in a heart biopsy, allows us to extract anywhere between 30 and 200 microgram of protein. So which is quite sufficient to perform proteome analysis and actually follow up experiments. So this is our routine workflow. And now I will come to the first data set. And I said, this is the first and sort of oldest data set that I raised from this, which motivated me highly to move on with this. And these are biopsies from TAVI patients. What is TAVI? Disease in this case is aortic valve stenosis. So the aortic valves basically are, when the heart is pumping, the valves in the heart are, have the job to maintain and build the pressure gradients. And that puts them under a lot of stress. So they age over time. So at some point they become leaky. So many people of you who may be in their 40s, 50s, 60s have probably gotten this message from the doctor that their valves are still working okay for their age. This is the message that I got at least. But in some cases, they can degenerate quite heavily, which means that at some points the pressure gradient that the heart builds during the pumping is no longer built properly, which means reduced blood flow and reduced performance of the heart as a pump, which also has knock-on effects. For example, the heart compensates for this by actually hypertrophy. Loss of function in heart disease is often compensated by hypertrophy, which if going for a long time can lead to heart failure. So this is a very widespread degenerative or wear and tear disease, cardiac disease. And one of the treatments nowadays, if it becomes really severe, you get an artificial valve implant. And this valve implant is actually done nowadays usually by a catheter operation. So it comes in a folded state by a catheter, which is going into your, don't know how to call it in English, where your legs start. And the catheter moves on up to the heart. And then when it reaches the proper place, first with another device, the old valve is actually cut out or cut away. And the new valve then unfolds the synthetic valve. And this operation is referred to as TAVI, or transcatheter aortic valve implant. And it's a standard therapy here in Germany for replacing leaky aortic valves. And being a university hospital, we have a sort of luxury that we have often the possibility to take small biopsies, not necessarily from the valve itself, but actually from the left ventricle opposite the valve. Because what we've come to realize is that it's not only degenerative disease of the valve, but actually the whole cardiac tissue around it also degenerates, which leads to the loss of function. So clinically, this manifests actually not as a single disease phenotype, but it actually 4 different phenotypes. This is a classification that is done by the American Heart Association, for example, the AHA. And basically, the manifestation of this loss of function of the aortic valve is present in 4 different subtypes: high flow, high gradient subtypes, low flow, low gradient subtypes, and then paradoxically low flow, low gradient subtypes, or subtype 4, which can be differentiated by a clinician based on echocardiography. So the clinician can actually make a judgment on which of these subtypes it is. And this can actually, or may lead in the future to different types of treatment. But we want to understand how these different subtypes come about. Why do we have different, say, losses of function of the ventricle and the valve, which lead, of course, to the same need for operation of a TAVI? But how do they come about, and how are they different on the molecular level? And the fact that they are different on the molecular level, you can see by these ear-stained biopsies samples from heart biopsies of these different subtypes. So you can see going from non-failing or sort of healthy human heart on the left, in the bottom row, you can see that you get quite some different morphological patterns in the tissue. So apparently different subtypes are manifestations of different degradations of the actual underlying tissue in the ventricle. So we decided to look into this on the proteome level and see if we get differences there. So what did we do is we took a smaller study, 5 versus 5 versus 5 versus 5 versus 5, so total 25 samples. from TAVI patients and also from some healthy hearts, which were operated for other reasons. And then we decided to look at these in two different fashions. The top level is we would take small biopsies from the left ventricle and analyze them by the workflow I just presented by proteomics mass spectrometry, which allowed us to get quantitative profiles for around 2,200, 2,300 proteins across all these 25 samples consistently, which we analyzed then by a relatively simple statistical analysis, so for differential abundance. And for proteins of interest, we also took aliquots of these biopsies then with proteins that showed up in the proteomics analysis, labeled them with antibodies, fluorescent antibodies for specific proteins, and then performed first confocal microscopy and then super resolution microscopy, or STED, to actually look at specific proteins and see if we could also see especially changes in the localization of these proteins across the functional heart tissue. Which may actually explain the different functionality of the heart tissue, which leads to the four different phenotypes. So this is a combination of mass spectrometry and super resolution microscopy, which proved to be very, very valuable in our case. So first we took the differentially abundant proteins and submitted them to an unbiased, unsupervised hierarchical clustering analysis. And what was extremely surprising is that this immediately reflected the phenomenological subtypes, the phenotypes of aortic valve stenosis. So you can see this clustering here on the left, the non-failing or NF heart. We have very clear clustering for the green and red samples, which is a low ejection fraction, low gradient, and paradoxically low gradient sample types. And we had a sort of slightly mixed clustering of the blue and yellow sample groups. But the differentially abundant proteins showed us very clearly that the phenotype correlates to a proteotype. So different types of failing heart tissue, or say partially dysfunctional heart tissue, actually are reflected both in a different phenotype of what is at top view the same disease, and different proteotypes. And we think that this may offer us some understanding on why these degeneration and this the need for replacing the aortic valves actually come about over time. And the same is reflected in a little less resolution actually here in the PCA, principal component analysis. So now you can go about and see, okay, what are actually the changes? What causes these changes? Which are the differentially abundant proteins that may relate to function? And this is here a scatter plot of both the low ejection fraction high gradient samples, so light blue, this on the y-axis, and the ratio of protein abundance to the non-failing heart. And on the x-axis, we have a different subtype of aortic valve stenosis. In red, the paradoxically low flow, low gradient phenotype samples, and their ratio to the non-failing heart. And you can see now that for a lot of these proteins, actually, you get the same trend, which is why we have the data points of the differentially abundant proteins on the diagonal here. And I've annotated here quite a number of these proteins, which are actually involved in the calcium influx or calcium balance. So the big calcium relevant channels like the ryanodine receptor or IIR2, SERCA2 are annotated here, and you can see that they're exactly on the diagonal, which means that they change the same way for these two disease subtypes. There are, however, also proteins which are off the diagonal, for example, the caveolin-1 or CAF1. And that's, for example, is an interesting one. Caveolin-1 is also has a big role in organizing these calcium balance complexes, transporter complexes across the membrane of the sacroendoplasmatic reticulum. But it is also increasingly implicated actually in signaling and organizing the mitochondria. and the respiratory chain. So the whole mechanism by which cardiomyocytes generate and also transfer their energy. So we think that we have here a good handle now by looking at the differentially abundant proteins, especially small differences that may allow us to get more insight into the disease processes of how aortic valve stenosis comes about a long time. As I said, the next part, which is not mass spec, but I still want to show you that because it's extremely nice. We looked at some of these of the underlying proteins. For example, here in protein stained in red on some of the biopsy samples called connexin 43 or CX43. And as a background here in green, you have a wheat germ agglutinin samples, or basically background structural staining. And if you look especially at the bottom, you can see that the CX43 stain shows that there's a significant CX43 loss, in this case across all 4 disease subtypes, compared to the non-failing or healthy heart. So, and what you can also see, if you look at the top row again, we can see very cleanly where actually this CX43 or other proteins involved in our data set actually localize or change in localization or change in co-localization. So here we have an extremely valuable tool that allows us to generate hypotheses on the proteome level by mass spectrometry about which proteins are involved in the disease processes in the human heart. And actually, by using the fluorescently labeled super resolution microscopy, we can also now in many cases actually link this to spatial distribution information, co-localization information. And this is something that we are following up on and which allows us in many cases to raise very, very interesting data. Now, where do we come now to resin, and why am I presenting some of this in this eMERGE seminar here now? Because we see a lot of promise actually for magnetic beads in helping us with this workflow. Here in the top row, I've basically outlined a little how we do this. The first 3 steps of tissue lysis, reduction and alkylation, and trypsin digestion, we can actually nicely automate in the barocycler device, in this pressure cycling technology device, which you saw. 16 in parallel takes around 4 hours total as a protocol, so that part runs pretty smoothly. The part which is much less smooth and actually less reproducible, as we found out, is the desalting, which initially we used basically C18 packed tips, stage tips for. And this is a highly manual approach, takes around 4 hours for a set of 16 samples if you do it manually. Of course, you can automate this by robotics. But for starters, we did this manually for a long time. You have to then speed vac the samples to dryness, which takes another 4 to 6 hours. But my main gripe actually with this reversed phase desalting is that it is actually the least reproducible step in the whole workflow, which we found out by doing serial measurements of aliquots. And then we started looking for alternatives to that. And one alternative, which we are moving towards now, is to use actually magnetic bead-based approaches. And this I've depicted here in the bottom row. And we would still do the tissue lysis with the pressure cycling technology in the barocycler, as you see, but then actually transfer the liquefied samples out and perform a magnetic bead-based SP3 type reduction, alkylation, and tryptic digestion of the samples. So what do? We bind the proteins coming in, from the lysate onto magnetic beads, onto the surface. We can wash away all buffer components in this SP3 approach, which was first published by Jeroen Krijsveld's group in Heidelberg, actually. And then we can make an on-bead tryptic digest, basically. And the nice thing is, if you optimize this nicely and reduce the buffer concentrations for the last step, for the tryptic digestion as much as possible, you can actually get away with the desalting. So if we use this magnetic bead-based workflow, the final step, the digest, the tryptic digest, we perform in 25 millimolar ammonium bicarbonate. And this allows us to get rid of the desorting step altogether. So the samples are mass spec ready, basically. And if we get the concentrations and the volumes right, we don't even need to reduce the volume in a speed vac or other, but can actually do directly DIA-MS analysis. And this DIA-MS analysis, also we changed the mass spec instrumentation to a Bruker timsTOF Pro in the meanwhile. Software changed to Spectronaut. And all of this has allowed us to increase the reproducibility of this workflow, which I hope to be able to show you in a minute. With the magnetic bead workflow, Stoyan and also others at Resent have been extremely helpful in establishing this for us. At the moment, we're using the Heilig beads for this. And another thing, this can also be automated on instruments like a Kingfisher, or basically any liquid handling device or automation device that can handle magnetic beads. And this is something that has good potential to accelerate our workflows here. But actually, even more important in the context of clinical research is to improve the reproducibility of things. So a short word on the timsTOF Pro, which is a more recent instrument. Ours dates from 2020, actually, where the mass spectrometry analysis or time of flight analysis is actually enhanced by both the pooling and the pre-separation or pre-fractionation of the ions by ion mobility in the gas phase. So in the interface region of this, it's manufactured by Bruker timsTOF Pro, you can not only enrich ions in the interface region. These ions of 4 different colors here reflect ions of 4 different m/z values, but you can also release them into the mass spectrometer one after the other. So in addition to separating ions by the m/z value, mass over charge, you actually can separate them by their ion mobility. And this combines extremely nicely with data-independent acquisition mass spectrometry. Because not only can we now isolate and co-fragment peptides in DIA by in mass over charge windows, but also in ion mobility windows, which you see here in the small red lines. And this allows us to get very or highly pure peptide populations in these windows. And because the instrument does this at great speed and sensitivity, this has greatly improved really the performance of the overall workflow. So how does this play out? Second application or second example I've brought to you is DIA-MS analysis of engineered heart muscle. How does this relate? Before I showed you something about actually human heart biopsies, but actually we are also looking here in Göttingen increasingly at therapeutic options for heart disease, for degenerative heart disease. And something which has raised a lot of attention recently is the use of engineered heart muscle. And I'm going to show you some data from our collaborators, the Vogt and Zimmermann labs here in Göttingen, actually, what they do. What they do is they co-cultivate cardiomyocytes with, for example, fibroblasts in special conditions, special media, which needed to be worked out across something like 2 weeks and also longer. And then you can see that actually from this co-cultivation forms tissue type entities, or engineered heart muscle, as they call them, which you see in the orange slide after 2 weeks. And if you do it right, actually, and get the conditions right and provide some stimulation, actually these loops of tissue, of pre-forming tissue, actually start also to contract. So they start to exhibit contractile function in frequency, which is very similar actually to that in the human heart at around 1 per second. In the middle, when you lyse this artificial tissue types affair and do cell sorting, you can see that you get a nice assortment of cardiomyocytes and fibroblasts, both healthy and viable, in red and green. Top right, you actually can see that this also scales up very nicely. So it responds, for example, to the calcium concentration. So calcium 2 plus is the regular stimulus of the heart for contractile function. And the force of contraction, FOC, over the cross-section, CSA, actually increases from week 2 to week 4 to week 8. So what you really have is slowly maturing and growing mimic of heart tissue. Bottom left, you can also see that if you do transcriptome analysis, this growing tissue is very interesting because it exhibits both partially expression patterns on the transcriptome level that mimic embryonic heart, so very early stage heart tissue, which is interesting because the embryonic heart can still regenerate. The adult heart tissue cannot regenerate well or at all. And some of the features of this EHM are also similar more to adult heart tissue. But what you can do is, and this is now the therapeutic approach, which is currently very, receives a lot of attention and goes into the first clinical studies now. You can provide small patches, like a fingernail size of patch, which is depicted in the F-actin immunostain at the bottom, centimeter size roughly, and slap them onto damaged heart tissue in an operation. And the interesting thing is the damaged heart tissue then starts to regain function. So it starts to increase function, contractile force again. It's not yet 100% clear how that works, if actually this tissue also starts to add its contractile function to the contractile function of the damaged heart, or if this is an endocrine effect. But the therapeutic usefulness is increasingly being recognized. So what we wanted to see now is, can we also do proteome analysis using our tissue-based workflow in this, say, sort of reduced but functional heart model of engineered heart muscle. So for this, we looked at, in an example, at EHM, engineered heart muscle under pacing stress. Pacing stress is a model for actually atrial fibrillation, where you put heart tissue under stress by applying it to either irregular or higher frequency electrical pulses, or optogenetic pulses can also be applied. For example, at 1 Hertz or even at 3 Hertz, which mimics actually the much increased heart rate of a fibrillating heart or irregularly beating heart. So what we did is we looked at engineered heart muscle, performed our tissue lysis, magnetic bead cleanup, mass spectrometry, DIA-MS workflow to look at what this does actually to the cardiomyocytes and the fibroblasts in these samples. So we took samples from these 3 different stages, control samples and samples of heart muscle paced at 1 Hertz and at 3 Hertz in collaboration with a group of Niels Vogt, who is doing a lot of very highly interesting clinical research in these models for atrial fibrillation. And we were able now to get consistent protein quantitation of up to 5,000 proteins, or protein groups rather. This actually corresponds to around 8,000 to 9,000 proteins. In brackets, you have the numbers which are consistent across 100% of the injections actually. So we have actually around 85% recovery of the quantitative values across each and every injection in this, say, medium-sized studies, which corresponds to up to 46,000 primary peptide sequences. The reproducibility was also quite good. Graphically, I think you can appreciate this from the total interferomatogram overlay, which I've put into the bottom. If you want to put this into numbers, if we do NS9 total, so 3 EHM replicates, tissue from 3 different EHMs per group, and also 3 mass spec replicates, we get in the range of 22, 23, 24% coefficient of variation, which is, allows us quite usefully some statistical power to look at differential protein abundance. And the whole thing for the small study is basically done using this straightforward DIA-MS workflow in 36 hours of mass spec time. So it's something, as I said, if you, going back to my initial comments on the Matthias Mann paper, Doll et al. From 2017, yes, that had the full detail, but took 3 months on 3 samples. here we can actually generate a lot of value for both this engineered heart muscle, but also for live or actual patient biopsies in a matter of days, basically. So if we look into this, what do we get? On the left side, you have the ranked abundance plot, where basically the proteins are ranked by the protein area, so the integrated and summed up area values of the peptides for each protein. You can see that we cover a range of roughly 5 orders of magnitude, linear dynamic range, all the way from 10 to the 7 to 10 to the 2, which reflects the challenge that we actually have when we analyze the proteome of heart tissue. Especially the cardiomyocytes have a huge dynamic range. Again, going back to the Matthias Mann presentation at the beginning, we have a lot of proteins in the contractile apparatus, and a lot of them are actually large, like titin or myosin or so. This is huge proteins at very high abundance, producing hundreds of triplet peptides upon digestion. So this is why cardiac research proteomics actually has a special challenge in that we really have a huge dynamic range, which much exceeds that of, for example, HEK or HeLa cell systems. Reproducibility is actually good. I've brought you here also the normalization plots. So the normalization of the quartile distributions of the protein intensities before normalization and after quartile normalization. So you can see we can do this or perform this whole workup at really high reproducibility. And then if we look at differentially abundant proteins, this is now simple student's t-test with Benjamini-Hochberg adjustment and so on. And now if you compare, for example, the 3 Hz pacing with our non-paced control EHM, you can see that we get quite a number of proteins differentially regulated after a few days of exposure to the pacing. And if you look at these, I cannot go at the moment into much detail with regard to individual proteins or biology, but if you do a set enrichment analysis, we see quite significant on statistically significant impact on lipid metabolism and on cell adhesion. And if we look at the cellular locations where the action takes place, to put it simply, we see a lot of changes in the extracellular matrix, so the organization of this engineered heart muscle, which you would expect, but specifically also a lot of changes in the mitochondria. And this is a pattern that emerges, that whatever we do to these engineered heart muscles, but also whatever heart disease type we are looking at, a lot of the disease effects play out on the level of energy production and energy transfer in the mitochondria, and specifically also in the mitochondrial inner membrane. So again, we get very useful data at high analytical depths of up to 5,000 proteins at the moment across many samples. So now, final thing, phosphoproteome analysis. If you have good model systems in hand and have a workflow for doing proteome analysis, why not go one step further and do phosphoproteome analysis? And here I would like to present to you some data that we raised together with the lab of Stefan Lehnert, cardiology research here in Göttingen, whom we have a very, very close and long-going collaboration with, and was done by a master student of mine, Dennis Olinik, who has now moved on to Jena here in Germany with Florian Meyer. And the reasoning was like this: if we have this nice workflow for abundance proteomics, global proteome profiling, why not go one step further? Why not do this here? So we take, again, our helic-based sp3 type workflow to isolate and digest proteins from the barocycler tissue lysis. At the moment, we still have to clean them up a little to reduce the impact of buffer using stage tips. But we are thinking about how to get away with this. And then we perform again an enrichment using magnetic beads. In this case, the Macrosin titanium IMAX HP beads that Dennis worked with. And then again do DIA and see if we can do phosphoproteomics by data independent acquisition. And the challenge in this is we want to do this at the moment in disease models, and most of the disease models in cardiac research are actually in mouse. Why is that a challenge? Because, sorry, go back one. Mouse tissue, mouse hearts are none too large. So from a left ventricle of a mouse, you can get around 200 micrograms of protein equivalent. If you compare this to the amounts which are often used for global phosphoproteomics studies, say in oncology cell lines, people often start there even with 5 to 10 milligrams of protein equivalent. So one of the big challenges here that we are facing is that we are trying to scale down a lot of the published workflows and so on by at least a factor of 10 or even 20. So this is what we are trying to achieve here, be able to work from either mouse hearts, or if we want to move, for example, to the analysis of the engineered heart muscle, to very small biopsy sizes, which only give us a few hundreds of micrograms of protein, and develop a very sensitive workflow for serial phosphoproteome analysis by DNA. So disease model in this case is phospholambanopathy. Here, a little background on this. Phospholamban is a small protein. It's actually around 53, 54 amino acids. Sequence is in top left, which has inhibitory region and a regulatory region. The inhibitory region is largely located in the SER membrane, so it's membrane spanning. And the regulatory region is out in the lumen. And the activity it inhibits is by controlling the activity of SERCA, of the big calcium pump, which largely impacts on calcium balance and therefore on excitability of cardiomyocytes. And to do this, to stimulate SERCA, actually phospholamban needs to be phosphorylated in a residue which is outlined in figure A top left by this arrow on the serine. This is a serine 16. And of course, to be phosphorylated there, we need a recognition motif for kinases. And this usually works out. The part when it does no longer work out, which makes this a disease actually, is when the arginine 2 steps to the left, which is the arginine 14, is actually deleted. And this is a mutation present in humans, but also in our mouse models, which is relatively rare, but it's regionally very widespread. So for example, there's pockets of this R14 deletion, for example, in large parts of the Netherlands, or in Greece, actually in Bulgaria. And also in the US, there are pockets where a lot of Dutch immigrants settled. So this is really something that is a strongly inheritable mutation, which basically is directly linked to a disease. How does the disease actually manifest? Many people develop loss of function of the heart, atrial fibrillation, and up to sudden cardiac death around the age of 30 to 40. And this is, so it's very disease, and actually has a relatively high incidence of disease of people who are otherwise in their most productive phase of life. If you want to learn more about this, I heavily encourage you to look at the Phospholamban Foundation's website, bottom right, en.plnheart.org, which is a foundation actually founded by a very nice individual, Peter Gleinis, who basically invested most of his personal finances, which unfortunately are not, or fortunately are not low, into research into this phospholambanopathy, because he is a carrier himself. So this is actually a very, very interesting disease model. And I can, as I said, only encourage you to look into this. So the mutation does the following. It abolishes the phosphorylation motif on the serine 16. So if this is abolished, less phosphorylation on the serine 16, it's heterozygous. Less phosphorylation means less SERCA2a activity, which means a long-term calcium disbalance in the cardiomyocytes. And this is how this disease, roughly speaking, comes about. So what did we do? We looked into a mouse model for this. There's a heterozygous mice coming to us from the University of Groningen in the Netherlands. We looked into a very early age, between 9 weeks in this case and 14 weeks, where we do not actually yet have a severe disease phenotype. So these mice are not actually close to dying or so. disease phenotype, which is severe, sets on at around 22 weeks of age. So this is really an early pre-phenotype, but which we think allows us to look closely at the actual beginnings of the disease. So Dennis took 6 mice each, healthy or wild type, and the phospholamban deletion mutant mice, isolated around 200 microgram of protein per left ventricle, and then performed the described workflow using the magnetic bead, both digestion and also the phosphopeptide capture. And this being cardiac research, not oncology, the numbers don't really compare to what many people are used to. Still, I think given the limited size of these biopsies, or sorry, of the heart pieces that we got, limited amounts of protein, and also especially the large dynamic range of the protein abundance in cardiomyocytes, I still think this is a very good first step, actually. So basically, he identified around 3,700 modified peptides, 1,300 quantitated by DIA across all injections. But ultimately, with some interpretation, we got around 3,000 modified peptides, phosphopeptides to work with. On the bottom right, you still see again the before and after normalization. But you can see even from the abundance distribution, the quartile distributions before normalization, you can appreciate, I hope, that the overall workflow is quite reproducible if you put your mind to it. And also, we are actually seeing differential changes in phosphorylation. And this is the volcano plot on the bottom left, on main part of this panel. And in small blue, which you can probably hardly see, this is unfortunately a screenshot. I didn't get around to enlarging this in time. I included this relatively last minute, but this is actually our controlled false correct positive. This is the phosphorylation sites on the serine 16 and on the neighboring threonine on the phospholamban. And we actually recover these at log-fold change of around 1 to 2, which doesn't sound like much, but think about this. This is a heterozygous mouse. So we're not expecting actually huge changes in the phosphorylation here. So what does it look like on the data level? This is exactly the example. This is the DIA data independent acquisition traces for the relevant phosphopeptides. We actually co-quantify here. We can sort of partially differentiate them. The phosphorylation on the serine 16 and on the threonine 17, which is right next to it. And in the middle traces, you can see the extracted iron chromatograms for the phospholamban deletion mutant mice. And in the bottom trace, the same for the wild type. And I think even on the raw data level, you can appreciate that we get quite significant and stable differences in phosphorylation for the relevant phosphopeptides between the wild type and the deletion mutant. So this is our, say, controlled true positive. And we are now looking into the other phosphosites and their changes. And also we are looking to improve this workflow again. This is so far as Dennis, a student, brought it during his master's thesis, which I thought was quite very good achievement. So coming to the summary, what I hope I've convinced you is that this DIA mass spectrometry approach is really suitable for serial proteome profiling, abundance profiling, but also with some more improvement in tweaking phosphoproteome profiling of cardiovascular tissues. Immediately, of course, for research purposes. We will see if maybe in the long run, this will also give us a lever on diagnostic approaches. We can do this on cardiac biopsies, like the first example from the TAVI, on cardiac tissues from model organisms, mouse, rat, and increasingly also pig as a large animal model are used widely. We can also do, which I haven't shown here, actually use this on vascular tissue. So for example, on the aorta itself, which is a little more challenging yet, and on engineered heart muscle, which is a very nice, both functional model for heart research research and also a strong therapeutic option. What are the key things here? Controlled and reproducible sampling. This is, I haven't talked about much, but it is a good idea at some point to make an appointment with your cardiology friends and ask them if you may not be able to sit in on an actual operation or so, or at least go with them very much in detail through the sample assevation steps. Because obviously when somebody undergoes a heart operation, even if it's just a catheter operation, asseverating samples for research is not the first or the second thought. So, but this is something that needs to be figured out, and due to our good collaborations here, close ties to our clinician friends, we have, I think, a good grip on this. The second, what is also key, is parallelized, automatable, and reproducible sample workflows. As said here, we are convinced, and also we have good evidence, and I hope I've showed you some good examples, that magnetic beads will take us here a long way to make this happen. And as said again, I would like to thank Stojan and team for their support on this. And finally, also we are trying to improve also the mass spec part of things. Being a proteomics lab, we are always technology oriented. So the combination of DIA with ion mobility separation, like in the timsTOF Pro instrument, has improved the performance of the whole workflow a lot. So coming to the end, thank you first for your attention. Some acknowledgements here are very much due to my team in the core facility Proteomics. Lisa Neuenroth is a fantastic technician, but also students, Dennis and also Hugo and Yannis, who are doing research on cardiac tissue. Definitely to a number of very good students in the lab of Stefan Lehnert here, cardiology research, translational cardiac research. Specifically Sören Brandenburg, the surgeon who provided the samples for our first study, two very good postdocs, Daniel Kovnatsky and Brian Fu. Other collaborators, the Vogt lab, who supplied the samples for the engineered heart muscle. Engineered heart muscle also needs a very strong stem cell unit in the background, which is led here by Lukas Zuganek. And I also have the luxury of receiving a lot of support by my academic boss, and who also leads the lab at my second association at the Max Planck for multidisciplinary sciences, Henning Urlaub. And with that, I would like to close and would be happy to answer questions if there are.
Thank you very much, Christof. This is super insightful. I really enjoyed it. So maybe I can get the ball rolling on a little bit of a discussion. For me, maybe the first thing to kind of focus on is one of the points you actually ended on, is the controlled and reproducible sampling. Mm-hmm. So I kind of have a few questions around it. One is, obviously heart tissue can degenerate quite fast. And so is there specifically ways to make sure that what you're analyzing is a viable tissue and you're not actually measuring artificial changes in the proteome induced by how the actual tissue was sampled or actually handled prior actual extraction?
Difficult to answer. I mean, this is a little like Schrödinger's cat. You cannot look into the box and actually have the cat live. Difficult to answer really is how close we are to the live tissue. Currently what we are doing is we are trying to standardize as much as possible the timeframes and the workflow together with the clinician. So it's always done quickly. It's ideally always done the same way. This is the most important. To have reproducibility in this case, I think is even more important than accuracy. So currently what we try to do is when the biopsy is taken, we try to ensure that the people in the operating theater asseverated already close in time after the same procedure. We do a few washes to remove excess blood, definitely, and then fresh freeze in liquid nitrogen. Currently, this is really the method of choice for the biopsy samples. It is still a good question. We are still looking at tissue which is sitting there at least on the time scale of minutes before this gets done. So a question which is really open is, for example, how useful it will be to do phosphoproteomics, for example. because kinase activity may go down after a while, but for sure phosphatase activity stays on for a longer time. So phosphoproteomics, or the viability of phosphoproteomics from live operation biopsies, we still have to establish, I have to say.
Yeah.
For mouse models or for the engineered heart muscle, that's a different game, of course. There we have much more control over time frame between sacrifice and so on. For example, if we sacrifice a mouse, that can be done relatively quickly. And then we can perfuse the heart, for example, with inhibitor cocktails. And there we think we are quite close. And for the engineered heart muscle, actually, you can do it in under a minute, basically, because that is only cutting and freezing.
Okay.
So that's pretty quick.
If you start seeing a lot of these stress-related pathways in your functional analysis, that might be a alarm bell as well. So related to this, maybe the question that's coming in is, because, I mean, the heart as an organ is so heterogeneous, you essentially, and you started this with the Matthias Mann work, is they sample different regions of the heart.
Mm-hmm.
So if you're doing a specific study, you would just focus on a specific subregion of the heart to be able to, you know, answer that particular pathological question, or would you sample across different regions to represent the different types of tissues and cells?
Again, difficult, not actually a differential answer to this question. Obviously, your options when it comes to heart biopsies from heart operations, your options are limited. Basically, you cannot pick and choose as you want. Basically, a lot of it is due to what is being done during the operation, where and how the catheter is placed, what the time frame is of things. Obviously, for this TAVI, the obvious place is to look in the left ventricle, and that's everything we have, basically. You know? Again, there the point is more to always do it the same fashion. And then actually we see from these biopsies, punch biopsies, as you saw, this is only NS5 per group, but still we got remarkably clean statistics out of this. Yeah. So as long as you do it the same way during the study, that is good, I think. The answer for mouse already is a different one, because we know, for example, this is relatively fresh information for this phospholambanopathies. Same as many other labs, we have started to look at the left ventricle because it has the most protein mass in murine heart and gives us the most to work with. Turns out, though, that actually the disease processes, which we know from other non-mass spec-based experiments, are actually more pronounced in the right ventricle. Yeah? So this is something really to think about, and why it's a good idea up front when you do this to basically catalog or look into the different compartments that you can. Maybe not all 16 like the Matthias Mann group did, but at least usually when we start working in mouse, human, we have already covered now quite some. We will soon invest into pig, actually, as a large animal model. You're well advised to actually at least catalog using your own lab workflows the different compartments that you may be looking into, because protein expression is compartment specific.
Yeah. The
answer for this engineered heart muscle is yet again different, because what is realized by transcriptomics is actually when as these heart muscles age from fresh human iPSCs over a matter of weeks, say 4 weeks, 20 weeks, the populations of cardiomyocytes start to differentiate. And this is something that is now realized by doing single-cell transcriptomics. When you do single-cell transcriptomics, you can actually differentiate and see that there's not just one type of cardiomyocytes, but based on who is doing it and who is doing the counting and the statistical analysis, between 6 and 8 different types of cardiomyocytes. So there's subpopulations. And the more fine-grained your view on this is, the single-cell proteomics currently is very educational in the field of cardiology research. The more you appreciate that we have different subpopulations of cardiomyocytes as the main players, and also these change over the course of maturation of such a synthetic tissue. Yeah. So there's a lot of work in the future to be done. I hope this answers your question about where you need to sample and how you need to sample.
Yeah, no, definitely it does. I mean, maybe a related question to this is, do you see difference in the extraction efficiencies if you sample different types of regions of the heart, or the methodology is kind of universal irrespective of what you're trying to extract?
I mean, the most qualified answer I can give you on that is actually from the human heart biopsies, because we have taken biopsies from the, I said these punch biopsies from left ventricle. And we have also done quite some work on samples taken during atrial fibrillation. During atrial fibrillation operations, there's a part in the heart, in the atrium, where the blood becomes stagnant. It's called, in German, it's called the atrial ear. And there, some blood settles, and also the tissue there degenerates a little quicker. And what is often done, this is removed by scraping it out, basically, which is a sampling technique which is different from a punch biopsy, because you're scraping more the surface of the tissue. And there we saw a lot higher variability. However, the yield of protein extraction actually remains similar. We are getting nowadays, when we take comparable volumes of heart tissue, to go by volume or by wet weight, we get a relatively stable yield of 80 to 150 microgram protein equivalent, which still is a factor of 2, but there's always enough to play with.
Yeah, definitely, especially with the new technologies in the back end. Okay, I don't see any more questions. Maybe I have one more from my side. In terms of the wide dynamic range that you mentioned in cardiomyocytes, are you doing any type of deeper diving to build your libraries fractionation wise, or going single shots at the moment? And maybe just related to this is, maybe I missed it in your presentation, are you using direct GIA now, or are you still kind of building libraries the traditional way?
Yeah, for library strategy, we are currently, because we have appreciated this localization of protein expression more and more, we are currently in the process of raising spectral libraries for our instrumentation done with our workflows, because the best libraries at the moment for DIA are still project-specific ones. From different compartments of the heart. I said not the 16 that Matthias Mann did to get comprehensive, but from all relevant parts which we might in the future get samples for analysis for, from left and right ventricle, from left atrium, from the septum, from the bottom of the heart. Not sure how that's called in English. Heart or base of the heart. We are raising location-specific spectral libraries. And at the moment, we are getting the best results if we take these location-specific libraries and combine it with actually identifications from direct DIA. So we are using what is called a hybrid library approach. If you use the terminology used by Biognosis, so the programmers of Spectronaut. So we use hybrid libraries, in other words. Project-specific, we take a pre-recorded spectral library where we do fractionation with of the peptide on the peptide level with reversed phase at basic pH. The actual DIA analysis themselves are single shots currently. What we are thinking also of extending, it's a balance of acquisition time and basically based on project time. You could think about a simple but hopefully reproducible pre-fractionation. Maybe also by magnetic beads or anything that can be automated nicely. You could also think, of course, of making longer gradients. This is always a balance between analytical depth and feasibility. Obviously, if you just do, say, 3 versus 3 versus 3, you can usually invest a good amount of mass spec time into that. Where we want to go is rather something like 50 versus 50 versus 50. And then you have to think about what is actually feasible with regards to the amount of instrument time that you invest.
Yeah, absolutely. There's always this playoff that you're going to have. Okay, I don't see any more questions. So I want to thank everyone for joining us and being part of this today's webinar. and especially to you, Christof. It was really great to hear more in depth about your research, and also from the biology side. It was really interesting, not just the analytical side. So thank you very much, and yeah, chat soon.
Yeah, thank you.
Cheers, everyone. Have a nice evening.